So in this lecture, I'll be talking about DNA sequencing. Okay. So first, let's go over some definitions here. So what is DNA sequencing? So basically, DNA sequencing is the process of knowing the exact order of nucleotides in a genome or in a DNA fragment. So basically, I give you a DNA fragment or a whole genome, and I ask you what is the sequence of this DNA. So you take it from 5' end to 3' end, and you start reading the DNA sequence going like A, G, C, T, A, T, T, whatever. Okay, all the way to the end. So that's DNA sequencing, and the purposes of knowing the DNA sequence is basically to identify genes and identify where they are localized. So you tell me that a gene X is located on this arm of the chromosome. It's near the telomere, near the centromere, in the middle, and there are certain numbers that indicate where genes are localized, and it is next to this gene or that gene, and so on. By knowing the DNA sequence, we can identify protein structure and function. So if we identify a gene, we can identify the codons, and we can translate ourselves the codons to amino acids, so we would be able to know the sequence of the proteins. And by knowing the sequence of the protein, we can predict what the structure of this protein is. And there are recently artificial intelligence and bioinformatics tools that can successfully, with high accuracy, predict protein structure. And by knowing the protein structure and the sequence of the protein, we can also predict the function of this protein, as well as other things like localization, what proteins it can interact with, and so on. By knowing the DNA sequence, we can also identify DNA mutations. So we have databases right now for what a normal human genome should look like. Okay, and we can compare the sequence of our unknown DNA or DNA from someone with a certain disease. We can compare it to this database, and we can pinpoint exactly where a mutation occurs and how it is related to a certain disease. We can also sort of like elucidate, clarify the genetic variations among individuals, among populations, in health and disease. So by knowing the sequence of the DNA of an individual, we would be able to know how variable it is in comparison to other or with other individuals. We would be able to predict the disease susceptibility of an individual. So by knowing the DNA sequence, we can say, you know what, there is a 50% chance that you might get this disease as you grow older, or that disease, or pay attention, don't take this medication because you have a genetic variant. Okay, you have this nucleotide right here in this gene, and that makes you a slow metabolizer for this medication or that medication, and so on. So we will be able to help an individual by letting him know to take care of himself or herself. Okay, we can also determine the best treatment for an individual, or if we, for example, if we know the sequence of the cancer of a certain person, we would be able to determine what treatment is best for that cancer. Of course, we can also determine how organisms are related to each other in terms of the evolutionary tree, let's say. Okay, so we'll be able to compare human genes with mouse genes or dog genes or cat genes or whatever, and this might help us understand our cells, our diseases, and how cells function normally or whenever there is a mutation. Because we use animals as model systems for humans. So these are the benefits of knowing the DNA sequence. Now, brief history, DNA sequences, or determining the sequence of genomes, of course, it started with simple organisms, simple genomes like viral genomes, bacterial genomes, because they were small, easy to handle. Then mitochondrial DNA was sequenced, followed by simple eukaryotes like yeast, the yeast cell, model system, Saccharomyces cerevisiae. Then the genomes of multicellular organisms was sequenced as well, like the C. elegans, which is a worm. And we learned a lot from this worm. Then, of course, along the way, the human genome was sequenced, and as I said before, it hasn't been completed yet. We still have to determine the final regions, the final pieces of the Y chromosome. But who cares about men? All right, so this table this is not for you to memorize, but this just shows a comparison of the genomes of different organisms and number of genes, protein coding genes, and number of chromosomes. So you can see here the E. coli, it is composed of about 4.6 mega base pairs or 1,000 base pairs, or sorry, million base pairs. There are 4,300 genes, protein coding genes, and there's one chromosome. So you can compare this with the yeast model system. There are 16 chromosomes, okay, about 6,600 genes. Okay, compare this to, let's look down here, the fruit fly, for example. There are 8 chromosomes, even though it is more complex than yeast, but yeast has more chromosomes than the fruit fly. Number of genes, about 14,000. Now, if you compare the mouse genome with the human genome, almost similar numbers. Overall, similar numbers. Well, compare this to chimp and dog genomes, very close to human numbers as well. Okay. So you can look at these numbers and enjoy. Don't memorize. This shows here comparison of genomes of different organisms, starting with bacteria and ending with mammals, including humans. So you can see how variable genome sizes are in different species, and it's sort of like related to complexity of the organism, but not 100%. Okay, and there's a lot of variation. So again, this is not for you to memorize. Okay, so let me refresh your memories in terms of nucleotide structure. So this is a nucleotide. It has a sugar. That's a deoxy ribonucleotide. That's the sugar. This is the base, 3 phosphates. So this is the substrate for the DNA polymerase. Note that the ribose sugar is a deoxyribose because it's missing a hydroxyl group right here. Note that we here at carbon number 3, we have a hydroxyl group attached to it or conjugated to it. And this is carbon number 5, which is associated with a triphosphate group. So whenever DNA is synthesized, you have attachments of this phosphate with the hydroxyl group at the 3' end, right? So this is the 3' end. So you remember that, okay? So you have this phosphodiester bond formation between carbon number 3 with carbon number 5 in the incoming nucleotide. So you have this phosphodiester bond. Note that energy is needed, of course, and energy comes from the release of 2 phosphate groups, and they have the formation of the phosphodiester bond between the phosphate and the hydroxyl group right here. Okay, so and it continues downward, or it continues from the 3' end. So in order to synthesize DNA, what we need is a phosphate nucleotide, and we need a hydroxyl group at the 3' end. Good. So let's start with talking about the technique. Now, the technique of DNA sequencing originally was based on the use of a dideoxynucleotide. So the dideoxynucleotide basically is similar to the deoxynucleotide. That is, you have again the sugar, the base, 3 phosphate groups, and you have a 3' end right here with the hydroxyl group. Now this is a dideoxy because it has a deoxy carbon right here. It's missing a hydroxyl group, and there is another deoxy group right here as well. So it's missing an oxygen, the hydroxyl group. And this means that whenever this is added to the DNA, no other nucleotide can be added to the 3' end. No phosphodiester bond would be formed because there is no hydroxyl group. There is no reactive group. So DNA synthesis, as a result, stops at this point. So this would be the very last nucleotide that is added to DNA. And that is what's used for DNA sequencing. So the idea here is that in order to synthesize, in order to sequence a DNA, what we need is a primer, just one primer, because we're gonna only sequence just one strand, not both strands, just one strand. So we need a primer. Of course, we need a DNA polymerase. We need a primer because DNA polymerase cannot start DNA synthesis de novo, that is from scratch, right? And the primer is labeled with a radioisotope like phosphorus, for example, radioactive phosphorus. Of course, we also need the substrates for the DNA polymerase. That is the 4 dideoxynucleotides, dideoxy A, C, G, and T as well. Now, we also need, and we need a dideoxynucleotide. Okay, so we're going to have 4 reactions, 4 tubes. In each one of them, we're going to have a template, the template to be sequenced, a DNA polymerase, the 4 substrates, a primer, a single primer, and 1 dideoxynucleotide. That is, in other words, to tube number 1, we add dideoxy G. To tube number 2, we add dideoxy C. And then we add dideoxy T to the 3rd tube, and we add dideoxy A to the 4th tube. Okay, so the idea here is that DNA polymerase will start synthesizing DNA, but then if it adds a deoxyribonucleotide, it can continue. But if it adds a dideoxyribonucleotide, synthesis is terminated. It stops because no other nucleotide can be added to the deoxy 3' end of the DNA. All right, so what we do then is that we separate the DNA fragments that are formed from in each one of these tubes. We separate them through gel electrophoresis, and we will have different fragments, different lengths of the DNA fragments. I'll show you now how they are separated. And the DNA fragments, by the way, differ by 1 nucleotide. So we're going to have a sequence of nucleotides, and they would differ by 1 nucleotide. I'll show you now how. And then we can expose the gel to X-ray film, and we can detect the signal. That's if we use radioactivity, but no one uses radioactivity anymore because it's risky, and there's a much easier method right now. I'll talk about it, but I just want to show you how it works. So here's the idea here. So we have a template. So that's our template that we want to sequence. We want to know the order of the nucleotides. We use a primer. Of course, we know what the sequence of the primer is, right? And this primer is labeled with radioactive phosphorus. We add to the tube DNA polymerase. we add the 4 substrates, normal substrates, and then we add to each tube 1 dideoxy, like I said. So let's say that we add to this tube dideoxy GTP, and we add it in low concentration. Okay, so whenever the DNA polymerase, whenever it sees a C, it's gonna add a G, and it has 2 choices. It either adds a deoxy G or it adds a dideoxy G, and the higher probability is that it's gonna add a deoxy, normal deoxy G, because dideoxy G exists at low concentration. So it's a matter of probability. So it's more probable that it's gonna pick a deoxy G and adds it to the growing DNA. All right, so here let's say that we start DNA synthesis, right? There's a C. So let's say that we have, let's say, 1,000 DNA molecules, okay? So of template molecules. So here's a C. Now the DNA polymerase has 2 choices. It either adds a deoxy G or a dideoxy G. Since the dideoxy G exists at low concentrations, let's say that it adds this dideoxy G to 100 molecules. So we have 900 molecules having the normal G, and as a result, the DNA polymerase can continue the synthesis of DNA. But for 100 of them, since it adds dideoxy G to these 100, synthesis is terminated, right? So we're going to have a very short DNA fragment right here. So let's say for the other 900, DNA synthesis continues. So there's an A T is added, another A, T is added, and so on. G, a C is added, and so on. It continues because all of them are normal deoxy nucleotides. And then there's a C. Again, there's a probability that the polymerase would add a deoxy G or a dideoxy G. If it adds a deoxy G, synthesis continues. If it adds a dideoxy G, synthesis stops. So let's say that for 400 out of 900, we have the addition of a dideoxy C. Now we have synthesis is terminated for these 100 molecules. So we have 800 molecules left where the DNA polymerase can continue synthesis. Now it continues all the way until it reaches another C. It puts a G, again, probability, this or that. And if it adds a dideoxy, then synthesis stops. So let's say that it stops for another 100. Now we have 700 molecules left for the DNA polymerase to continue synthesis of DNA, and so on. So notice that we're going to have DNA fragments of different lengths. Now, okay, so we have 4 tubes right here, okay? In each one of them, we have the double-stranded DNA, the DNA that we want to sequence. There is a primer, and the primer is labeled, and we have the substrates for the enzyme, and we have the enzyme, of course, the DNA polymerase. So in each one of these tubes, we have, again, the enzyme, the template, the primer, and the substrates. But for each one of them, we would add 1 dideoxy substrate. So we have dideoxy ATP, dideoxy TTP, dideoxy CTP right here, dideoxy GTP. Right. So synthesis would start. So right here we have a T. So an A should be added right there. Now for these 3 tubes, synthesis would continue normally because there's only a deoxy A. But for this tube right here, where you have a dideoxy ATP and a deoxy ATP, again, the DNA polymerase would add this or that. And there's a higher probability that it would add a deoxy A. But for the if it adds a dideoxy A, then synthesis stops. So synthesis would stop with the addition of 1A right here, 1 dideoxy A. Now let's say that in this tube right here, this one right here, synthesis will continue because there is an A, so it adds a T, there's a C, it adds a G, and there is an A, it adds a T, there's a G, it adds a C, and there's a T. Now, again, we have a probability of adding this or that. If it adds a deoxy A, synthesis continues, but for some of them, for some of the DNA fragments, a dideoxy A is added, and that results in the termination of some of these fragments. Okay, so note that in each one of these tubes, we're going to have different fragments of different lengths. Now we take, same thing with the other tubes right here. So here we add the dideoxy TTP, so the fragment ends with a T. Okay, here the fragment ends with a C, and so on. Now notice that the primer is labeled, right? It gives us a signal. So we separate the DNA fragments in a gel using gel electrophoresis, and the fragments are separated according to size. Now this is the shortest DNA fragment right here. Okay, this is the second shortest. This is the third shortest. Okay, so these fragments are gonna move really fast. Now this one right here is the longest. This one here is the second longest, and so on. So we have the large fragments on top, we have the small fragments in the bottom right here. So and these differ by just one nucleotide, and the resolution of the gel is really high. So we can separate the DNA fragments based on size, even if they differ by one nucleotide. So right here we have in this tube, whenever we see a signal, we know that that it has an A. It ends with an A. So by going from bottom to top, we would be able to know the sequence of the newly synthesized DNA. Okay, and by knowing the newly synthesized DNA, we will be able to know the sequence of the template. Note that here we start at the 5' end because whenever the A is added, it's closer to the 5' end. Okay, so the sequence of the newly synthesized DNA would be A, T, G, T, C, A, okay, G, we have T, C, C, A, G. So that's 5' end to the 3' end. So that's how we know the sequence of the DNA. Okay, so these are again different results. This is the gel done for the synthesized DNA. Here again we start from the bottom at the 5' end. So the sequence here is basically T, G, C, G, 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 C, T, T, A, then we have T, then we have C, G, 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 then go there, T, C, T, then A, A. Of course, by switching the gel, sort of like, okay, so it's hypothetical, so we would be able to tell the sequence of the template, right, by reading from by switching the letters. So instead of A, we would have a T. Instead of T, we would have an A. Instead of C, we would have a G. Instead of G, we would have a C. And we start reading from top. Okay, so this is sort of like a hypothetical gel for the template. But you can again go there and say, that's an A, so that's T, T, A, G. Okay, and A, and so on, C, 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 and so on. Okay, so you would be able to know what the sequence of the template is by flipping the sequences. And remember that DNA is anti-parallel, right? Okay, so that's the idea behind it. So let's say that that we have a two bands that appear at the same level. So they have the same size. Okay, what does that mean? Okay, shouldn't there be just one band? Well, not necessarily true. You can have two bands having the same length. Remember, we are diploid, our cells are diploid. Okay, so we have two chromosomes. So whenever we take DNA from an individual and whenever we sequence the DNA of that individual, we are sequencing both chromosomes, the paternal and the maternal, right? So the DNA polymerase would synthesize, would use this as a template and this as a template, and it would read them at the same time. So you will have A right here and A right there. Okay, so let's say that it goes something like this. Okay, so the newly synthesized DNA: A, T, T, G, G. Okay, C, C. Okay, then A. Okay, so they would be exactly the same, except that there are genetic variants. There are differences in DNA sequences among individuals. So at a certain point, the DNA polymerase might read a T on one chromosome, but it can read a G on the other chromosome. I mean, these are polymorphisms, like single nucleotide polymorphisms. So it would read a T in here and a G right there, okay? But then it goes after that and it says C, C, G, G, A, okay? It would be identical, and then there's another variation. So you can have DNA fragments of the same length migrating together, but in different tubes. And what does that mean? This is polymorphism. It can be polymorphism, or it can be mutation in one chromosome, right? How do we differentiate between mutation and polymorphism? Well, if it exists in more than 1% of the population, it is a polymorphism. If it exists in less, then it is a mutation. Now, what would happen if we sequence a DNA of a normal individual and we see a band right there? But then if we take DNA from another individual with a certain disease, instead we see a band right there. What does that mean? It means that this person has a mutation on both chromosomes. Okay, so it goes from, let's say, from A to T. So the person is homozygous, right? But the person has a mutation on both chromosomes, again, or polymorphism, right? And that's how it is. And then we have to study if this genetic variant or if this mutation is pathogenic or not. So in this case right here, the individual is heterozygous, or let's say a carrier. It depends on the change, if it is polymorphism or a mutation. Okay, this person right here is homozygous. Okay. Now, well, working with radioactivity is not really friendly. It's hazardous. The individual working with radioactivity can have the radioactive phosphorus get into her body, induce mutations. So it is harmful. So scientists said, well, wait a second, why don't we use fluorescence instead of radioactivity? And that would make it less laborious, you know, working with radioactivity for tubes and then looking at each lane and trying to read the sequence of DNA is really laborious. So they said, why don't we use one, fluorescence, and two, why don't we make it automated? Why don't we let the computer read it, or whatever instrument? So they said, okay, that's great. What we're going to do is we're going to use fluorescent substrates. So instead, so in addition to the deoxy A, deoxy C, the deoxy substrates, deoxynucleotides, we're going to have dideoxy A, but it gives a red fluorescence. We're going to use dideoxy C, and it's going to give a blue fluorescence, and so on. Okay, so in a single tube, we're going to have the template, the primer, but in this case it's not labeled. We're going to have the substrates, of course, the DNA polymerase, and we're going to have the fluorescent substrates as well, and each one of them would give us a certain color. So the DNA fragment would be red, right? So right here in one single tube, the DNA polymerase would read a G and add a C. It can add a dideoxy C, right? So and synthesis would stop for some fragments, but for the majority it would continue. So it's going to have a and there's an A, so it's going to add a deoxy T or a dideoxy T, right? Again, for some of them, but not everything. So synthesis would continue for the majority, and some of them would be stuck at the dideoxy T. Now remember that if we add a dideoxy C, now this fragment would fluoresce with a blue color. If there is if there are fragments having that, if there are fragments that end with a dideoxy T, these fragments would fluoresce with a yellow color. Okay. Now the fragment that ends with a T and the fragment that ends with a C, they differ by just one nucleotide. So we can have our sample then separate, we can have our DNA fragments separated through a gel, and they would be separated according to size, and the resolution is really high. Fragments can be separated by one nucleotide. So instead of having 4 lanes, we're going to have our samples separated, our DNA fragments separated in one lane. These are the large molecules, these are the small DNA fragments, and each one of them gives a certain color. And as they migrate, you have a sensor that reads the fluorescence, and it transforms the results in the shape of peaks. So it reads a peak right here, and it can translate it into a letter, any one of these, any of the 4 letters. So it would say, aha, this is this color, then it must be a C. This is that color, it must be a G. This is this color, it must be something else. So we have an instrument now that reads the DNA sequence, or the DNA sequence, by translating colors into letters. So we're gonna have results that look like this. Let's say that this is normal and this is DNA from an affected individual. So you look at the peaks right here, and you look at them and you say, and the instrument would say, that's a C, that's a T, that's an A. So again, it translates colors into letters. Now let's say that an individual has 2 peaks appearing at the same location. Now the instrument would say, oh wow, wait a second, we have 2 peaks right here, so there is a polymorphism or mutation, we don't know. So this person right here is heterozygous, because you have 2 bands that migrate at the same, you have 2 bands that overlap, and they give signals. So the instrument would read this signal and that signal at the same time from the same band. So it puts the 2 peaks at the same at the same position, and it gives us a letter. But we look at it and we say, wait, there are 2 peaks right here, so there must be, on one chromosome there must be a C, on the other chromosome there must be an A. So polymorphism or mutation. So we say that this person is heterozygous. Now, if the whole color changes, we expect it to be a T, red, but then it turns out to be blue, that is a C. This means that we have a homozygous individual, and it has a C on both chromosomes instead of having a T on both chromosomes. So we can say again, the person is homozygous, and but homozygous for a T or homozygous for a C, and so on. Now, things move really fast, and scientists said, you know what, why don't we make it faster? Remember that the Human Genome Project cost about $3 billion, and it took them, they're not done yet. They still have the Y chromosome to finish, and it takes a lot of time. So scientists uh, invented much faster method to sequence DNA, and they called it next generation sequencing. And there are different technologies invented so far, but we're going to talk about one of them only, which is one of the best um, technologies. So basically what happens is that a DNA is taken, and now it costs about $100 to $500, by the way. So that's how it goes. You have a DNA fragment, long DNA fragment that we want to sequence, a whole genome of an individual. We take this DNA and it gets fragmented randomly. So we have different fragments of different lengths, long and short, but usually they have different certain sizes or range of sizes. And since we don't have just one molecule, we have a lot of we have like millions of molecules of a human genome, and they are fragmented randomly. Some of these fragments would overlap, right? Because it's random chopping of DNA. So you will have fragments that overlap with each other. Now each one of these fragments is taken and an adapter is added. So DNA adapter is added. Pieces of DNA are added to the ends, to both ends of each fragment. Remember that they overlap, but these, the adapters, the DNA adapters, the DNA pieces that are added to the ends have known sequences. Okay? Now, then these DNA fragments are attached to a platform, to a solid platform. And we have DNA synthesis going on each one of these, for each one of these DNA fragments. Now, and we use the same primer. Well, how come they have different sequences? We use the same primer because they all have the same adapter, and the primer is complementary to the adapter. So we have sequencing going on for each one of these DNA fragments. Okay, they all have the same primer. Well, this platform right here can have billions of clusters, billions of DNA fragments attached to the solid platform. Well, how can we sequence all of these billion fragments at the same time? Well, that's technology. You have a camera that records what each one of these clusters does, what each color is given by each one of these clusters. So let's say we take one of these clusters and we add nucleotides. Now these nucleotides are special nucleotides. One, whenever they are added, no other nucleotide can be added. That's one. Two, in order to add another nucleotide, the nucleotide that is added must be modified, and then the other nucleotide can be added and the DNA polymerase can put it in place. But no other nucleotide can be added again. So it has to be activated. It has to be chemically modified. That's one. Two, each nucleotide gives a certain color. Okay? So whenever you have the addition of a nucleotide, it gives a color, and this color is recorded and it gets translated into a letter. And then it gets chemically modified so we can do another round, another addition of a nucleotide. So one nucleotide is added at a time to each one of these DNA fragments in a cluster. Okay, and we have billions of clusters. So we have another cycle right here. The nucleotide is activated, so we can add another nucleotide. A color is given, the nucleotide is activated, okay, and then we add another nucleotide, third nucleotide, a color is given, and then it gets activated, and we add the fourth one, and the fifth, the sixth, and so on. I wanted to watch the video at the end of this presentation. Now, so let's say that we have, this is part of the solid platform, slide, or whatever. So we have round one. Let's say that we add the nucleotides. To this cluster, a G is added. To this cluster, a C is added. To this cluster, a T is added. To this cluster, an A is added. And each one of them gives us a certain color. And we have a camera on top. And this camera detects the color that comes out from this cluster, and the color that comes out of that cluster, and the color that comes out from this cluster, and so on. Okay, so it tells the computer, in this cluster there's a G, in this cluster there's a C, in this cluster there's a T, in this cluster there's an A. Second round, these nucleotides are activated, they're modified, so we can add another nucleotide. So in this cluster you have a C added, yeah, in this cluster you have an A, in this cluster you have a T, in this cluster you have a G, and the camera reads these clusters one by one, or actually it does all at once. The third round, you have different colors generated from each cluster, and the camera reads them. So the sequence of the DNA in this cluster is G, C, T, G, A. The sequence of DNA fragment in this cluster is C, T, T, A, G, and so on. So we would be able to know what the sequence of each cluster of DNA. And it would really look like fireworks going on all at the same time. This is a slide 100 micron. This is only small part of the solid platform. Amazing technology. All clusters can be read at the same time by the camera, and each color for each cluster is translated into a letter. What we do then is that we can, using bioinformatics, we would take the sequences of each cluster, and since there's an overlap between the different fragments, the computer program, whatever, it can sort of like, it puts a sequence to all DNA fragments by combining all of this information. Please watch this video. You can watch it on the slide itself, or you can go to this link on YouTube and watch it, please. I have to warn you that the video contains a lot of information, okay? Don't worry about the itty-bitty details that exist in this video. I just want you to know the overall concept of DNA sequencing and the technology associated with this technology. So don't worry about the details, okay? Allah ma'akum.